welcome everyone. Excited to be opening the StatGen tutorials. I always used to be very jealous of people who would have very pretty pictures for their intro slide, and now there's generative AI, so I can have funky pictures too. So if you put my title into DALL-E, these are some of the first 4 images you get. I really like this one on the right, so I guess this character is presumably Pyro. And I think he's teaching this little blue guy about probabilistic programming. So you're all the little blue guy, and I'm the dragon thing. So I'm pretty happy about that. I put together a Google Colab notebook, which you can get to at this link if you so desire. So unlike other talks, you're very welcome to have your laptop out if you want to play around with the code. For those of you who don't know, Colab notebook is nice in that it just runs in your browser, 0 setup, so don't need to install anything. Also, no pressure. Give people a second in case they want this. So this talk is going to be about Pyro, and I will get to what that is. But I wanted to have a real problem. Yes, too soon. Yeah.
Thanks.
Yeah. Ideally, I could write it here, right? Actually.
I thought you posted on the Hoover as well.
I did post it on Hoover as well. That's true. Yeah, if you go to the information for this talk, it's posted in the chat, I think, right? Yeah. Yeah. OK, anyone need more time typing this? OK. So I wanted to have a real-world example of where we actually use Pyro for something rather than just a toy. And so I have a little bit of intro to get to that. So my lab, like many labs who will be represented at CGSI, is interested in understanding how variation at the DNA level ends up manifesting in traits like human genetic disease. We know this must happen through some mediators. One of the mediators that we're particularly interested in is RNA splicing. So this is where you get variation in which regions are removed from the RNA that's initially transcribed. And that variation, we know, can result in disease sometimes. Some of the work that I did actually during my postdoc was how to quantify RNA splicing from RNA sequencing data using this leafcutter method. I won't go through the details here because it's not very important for the point of view of this talk. But leafcutter has been used to find lots of splicing QTLs, for example, by GTEC. So this is statistical associations between genetic variation and RNA splicing. And what's really important about this is it turns out that you see really strong enrichment of those variants in many different GWAS. So it seems like a lot of genetic variation is having an effect on complex traits, in this case multiple sclerosis, is having that effect through an effect on RNA splicing. And so that gives you some idea that this variation in splicing is functionally important. One of the other ways we can get a hint that splicing is important is by looking at differences in splicing patterns across different human tissues. So the rows here are different introns, how much they're used, and the columns are different samples from different human tissues. You see some amount of conservation when you look across different mammalian species. Conservation of those differences in splicing between different tissues. So that's another hint that this splicing changes are functionally important. But we don't have much good experimental data genome-wide that tells us what's really functional. And for gene expression changes, there's been all this development of CRISPR-based perturbation technologies that can tell you in a genome-wide way what genes are having an effect on some functional output. So one of the projects my lab's been working on is trying to develop CRISPR-based approaches to screen for the function of specific splice isoforms. So the reason we can do this now is that there are CRISPR systems which target the RNA rather than the DNA. So the CRISPR-Cas systems you've mostly heard of are Cas9-based, which cut DNA. There are all these different Cas13-based systems now that target RNA. So they're cutting the RNA, not the DNA. So you can get a gene expression knockdown. And what we've been trying to do is figure out how to, if you target bits of the RNA that is specific to one splice isoform versus another, then you can knock down just one isoform and not the other. And then you can see if there is a difference between knocking down the 2 isoforms. So the challenge in doing this is trying to figure out, like, well, so I don't know how much people know about CRISPR, but you basically, the way that the knockdown works is you need the Cas protein itself, and then you need these little guide RNAs that you put into the cells that complementary base pair to the sequence that you're trying to target. And so one of the big challenges with all of these CRISPR-based approaches is to design these short guide RNAs that work well. And it turns out if you just design them at random, a lot of them don't work very well, especially for Cas13. So we did an experiment where we designed these gRNAs, guide RNAs, to overlap different positions over these exon-exon junctions. So this is where, from these isoforms, you get where so for example, in this isoform, we have this pink and this blue exon together, and we can design guides that specifically target the combination of those two exons. And one of the really powerful things about CRISPR is you can do these CRISPR screens. So rather than doing an experiment where you put in one guide RNA, you can design an experiment such that each cell is getting, usually, there's some trickery to get this to work out exactly, but usually one cell is just getting one guide RNA. So if you're talking about genes, then usually that means you'd be knocking down or knocking out this one gene. We're talking about isoforms, so we'd be knocking down or knocking out, or knocking down, in this case, one isoform. So the way these CRISPR screens work is you put in a big pool of these guide RNAs. In our setting, that's going to end up being around 50,000 different guide RNAs, so targeting different positions in essential genes. And the reason that you target essential genes is that if the guide actually works, then as the cells try to grow, if a specific cell has received a guide RNA that effectively knocks down the expression of an essential gene, then those cells won't grow, or they'll actually die. And so what happens is you run the screen, you start off with what's shown here, ideally like a nice even distribution of the different guide RNAs in your pool of cells. And as time goes by, some of those drop out. And these ones that are no longer represented or represented less are the ones that were effectively knocking down the expression of essential genes. And so that tells you both, okay, this gene was essential, and then also this guide RNA was effective in knocking down its expression. This is obviously, you know, a cartoon version of this, because this is only showing this for 6 guide RNAs. In reality, you know, here we were putting in 50,000 different guide RNAs. People do this up to sort of hundreds of thousands even. So what does the data actually look like once you get it back? So this is the data just for one specific guide RNA targeting one junction in an essential ribosomal gene called TSR1. And this is that data just plotted. So the experiment we did, we had 2 replicates. So that's basically doing the experiment twice. And we have time points at different weeks. So week 1, 2, and 3 was the final endpoint. And the y-axis here is log to fold change. So this is how much did the representation of that guide go down compared to the starting time point. So there's a little bit of normalization that's already gone into getting this y-axis value. I won't go into the details of that because I don't have time. So what you can see, though, is this fold change. So this fold change of 2.5 for this point, say, that means that at week 1, that guide RNA is whatever 2 to the power of minus 2.5. So something like 0.2, I think. So it's the amount that it's represented has gone down by that amount. And so, and you see that this keeps going, the trend is that this keeps going down in how much this guide is represented as more time goes by, the longer we grow the cells. So this is a guide that looks like it's working really well, but you can see there's also like a lot of noise, right? Like between these 2 replicates at the week 2, there's like a really big difference, and then they do seem to converge, but figuring out like actually how good should we think, how well should we think this guide worked, it's a little tricky, right? There's clearly some signal here, but also there's not many data points. And so that sort of motivates trying to be Bayesian and careful about uncertainty in terms of quantifying how well this actually worked. So we're going to be Bayesian, which means we're going to think about all the uncertainty in our data. And to give you a very quick Bayesian statistics 101, the fundamental idea in Bayesian stats is that you assume your data, D, was generated by some stochastic process Q, which we normally call a model, and that Q has some parameters theta. So we write that like this. And the key thing we're normally sort of the core of Bayesian stats is saying, what do I know about these parameters theta having observed some data set D? And that we get through Bayes' rule, which says that the object we should be interested in is a thing called a posterior. So this is a distribution over our parameters theta, conditional on having observed some data set D. And we get that through Bayes' rule, which says that what we need to do to get this posterior, we multiply together a likelihood. So this is more like the object we deal with in frequentist statistics, which says how likely is a given observation of the data set given some specific setting of the parameters theta. And then we have a prior. And picking the prior is always a little bit of a tricky thing, but I'll talk about that a bit. And then this evidence object, which is not going to be too important for now. Okay, so I'm not used to showing code in talks, but I'm going to show code to try and show how we can use Pyro to put together a simple model for the data that I just showed you. So the inputs here, so this model has some inputs which are actually just the data that are going to be, so the week is effectively, is that x-axis. And this is effectively going to be like a little regression model. The observed log fold change, that can optionally be none, which means we just run this model without conditioning it on a specific set of data. But we can also put in values there. And the first interesting part of the model is going to be, we're going to put a prior on this guide score. So this is really just the slope of that. So if you imagine putting a linear regression line through here, the guide score is just going to be the slope. That's telling us, because a significant negative slope here would mean that the guide actually worked. And the way you do this in Pyro, I guess I skipped saying what Pyro is. So the point of this is we're going to be able to do this Bayesian statistics analysis of getting that posterior object by writing down a model, that Q from the previous slide, and then Pyro is going to do the work for us of conditioning on data and giving us a posterior distribution. So the way we set up the model, we first need to specify a prior for our, in this case, we just have this one parameter, this guide score. And the semantics are you need to, this pyro.sample thing, you need to give it a name. There are some competitors to Pyro around where you don't have to actually explicitly give it a name, but they have other downsides. And then this dis.normal is just saying we're putting a normal distribution with, this is called a location, so this is just like the mean, and a scale. So this is like the standard deviation. I'm just putting some large number so that the prior is not very informative. Then this is just a very simple linear regression because we don't even have an intercept term, because from our normalization, we know that line should go through 0 at 0. And so we could, in principle, have had any PyTorch code here. So PyTorch, for those of you who don't know, is one of the, I think probably the most widely used deep learning framework these days. This is certainly not a deep neural network in any way, but we could actually have a much more complex function here if we wanted something like a neural network. But for now, this is just a linear regression, which says that the predicted fold change is the slope times the week, which is just the x-axis on that plot. OK, and then the likelihood. So this with pyro.plate, this is really just like a for loop, is the simplest way to think of this. So this is saying I have a for loop over the observations. So that's my 6 observations from that scatter plot. And for each of those observations, I'm saying that they are, again, I'm sampling from a normal distribution centered at this predicted log-fold change, and then with some variation, so noise around that center, and for now we'll just set that arbitrarily to 0.1. And this is where this observed log-fold change that we can feed to this function can go in, and that means that we're conditioning that the output of this sampling statement would be equal to this observed log-fold change. We can return all of these objects from the function if we want. You don't actually need to, but it's nice just, and we can actually use this function as a regular Python function if we want. So that's what I'm showing here. So one of the sort of slight caveats with Pyro is it builds on top of Torch, and so your data all needs to be Torch objects or Torch tensors. So you can't just stick in NumPy vectors or arrays. So you do have to do a little bit of this conversion, but it's not too bad. And then if we just call this function like it's a regular function in Python, and we give it, this is effectively our x, right? And this, we say we don't know what if we pretend we don't know what the observed log-fold changes would be, then we can just call this function and we get some data out of it. And this is what the data from doing that once looked like. Obviously, it looks nothing like the actual real data we had. This has a positive slope. And that's because if we just call this model without having trained it, then we're just sampling from the prior. So there's no information yet. So we want to fit our model to our actual observed data. And the default way that that's done in Pyro is using this process called stochastic variational inference. I'm not going to go into the math of this in any detail at all. I just want to give you the intuition, which is that what SVI tries to do is find an approximate posterior distribution. So something simple. In all of the settings I'm going to show, this is just going to be a Gaussian distribution. And we're going to try and find a simple posterior distribution, approximate distribution, that is in some sense close to this true posterior distribution that we would like to be able to estimate, but in practice is usually too complex. And the sense in which this is close is this distance called the Kullback-Leibler divergence or KL divergence. You don't really need to know too much about that, but I did want to mention that there's actually a whole family of these divergences that have different behaviors. The KL divergence itself, the picture here is meant to be this P is meant to be the posterior, so this is this slightly complicated thing with 2 modes. And then this Q distribution is this simple Gaussian that's trying to fit it. And you get slightly different behavior in how that fitting works for different possible divergences that you could use. The way that variational inference actually makes these things close is by this bit of mathematical trickery that tells you that if you maximize the thing called the elbow, which is a lower bound on that evidence object, then that turns out to be mathematically equivalent to making these things as close as possible in KL divergence. Not going to go into the math any more than that, because part of the point of Pyro is you can kind of not worry about the math and let Pyro deal with it for you. So to set up SVI in Pyro, you first need to say what your approximate variational posterior is going to be. And so one of the nice things about Pyro is it provides these automatic posteriors objects. This auto-diagonal normal just means that you're going to have a diagonal multivariate normal distribution on all of your parameters, and that's going to be your posterior, your approximate posterior. In our case, we just have one parameter, that guide score, so it's actually just a univariate normal. And then we're doing optimization, and you can choose any optimizer you like, but the most common thing is to use Adam, which is the most, probably, I think, widely used optimizer for deep learning stuff, which you set up very straightforwardly like this. And then you set up this SVI object that needs to know what's your model, what's your approximate variational posterior representation going to be. And then you do have to specify that the thing that you're optimizing is this elbow, so you can put in other options here. I think in reality, everyone just uses elbow. Then training this model, if anyone who's ever done any deep learning, looks very similar. You just loop through some number of epochs. Here, you can do mini batch like you would in deep learning. I'm not doing that here. We only have 6 data points anyway for now. And so you just do these SVI steps, and we can keep track of the loss and make sure that there's stochasticity in this, which is why you get this noise. But over the epochs, it goes down and eventually converges to somewhere. Once you fit your model, then you can extract the parameters in various different ways. So if you put dot median on the end of your variational posterior after doing that model fitting, then it will give you from the posterior distribution, the median of that posterior distribution for each of your parameters. In this case, we just have the one parameter, which is estimated as minus 2.2. Makes sense that it's negative because we saw that slope was negative. You can also extract the location and scale, so effectively the mean and the standard deviation of the posterior distribution. And so you can see, first one's not right, not sure what happened there. Anyway, you can see you can get a uncertainty measure as well. You can also do this by, instead of doing these sort of deterministic things, you can also just sample from the variational posterior. In this case, I'm sampling 500 points, but it's just a Gaussian, so you're not gaining very much information by sampling versus just getting the mean and the variance. So this is showing, if I sample from the posterior 500 times, I get there's a lot of little gray lines here, so you can see the spread, sort of the uncertainty the model has in the fit of this, and then the red line is the mean of the posterior, the fit that you get with that. So one other nice thing you can do, since we have a generative model, is you can simulate from the model, now that we've fit the model, and see if the data that you get from that simulated model matches what you see in the real data. So the only thing you had to do here in this predictive function was change the return sites to the sampled log fold changes. You can then plot what the data looks like when you do this. So all of these little gray points that are blurred on top of each other are simulated data from having fit this model. And you can see a bit of a problem already, right? Like the simulated data would all be very close to this line, whereas the actual data that we see is way further from this line. So we're clearly not representing the uncertainty in the data very well. So one limitation that you might think might be driving this is that we didn't learn what the noise was. So we fit a regression, but we didn't learn what the noise variance was, and that can be very problematic. And it's very easy. I think one of the takeaways I want you to have from this is when you make extensions to the basic model, it's very easy to do that, to add these little changes, to say, okay, well, I had that model, but the thing I want to change now is I actually want to learn what the variance of the noise is. So to do that, I need a new parameter, which is the noise scale. So this, if you wanted to think in variances, this is just the square root of the variance. You need to pick a prior. This comes from the Stan folks, people who know Stan. It's another probabilistic programming language. And they often recommend using this half-Cauchy distribution, which is relatively uninformative. So it lets the data speak for itself. And once we've put a prior and introduced this noise scale parameter, then all we need to do in our likelihood is to say, if you remember before I had the scale being equal to 0.1, now the scale here of the noise is equal to this noise scale parameter. The inference code doesn't change at all. The only thing I've added here, and you'll see this in the notebook if you look at it, is I'm initializing the guide score parameter estimate to what it was from the previous fit. One of the common ways of making things work a bit better in these Bayesian models in general, but in Pyro in particular, is to give reasonable initializations to the model fits. But the rest of the code is exactly the same. And so we can again extract the estimates from this model. And the nice thing now is that if we simulate data, now we've learned the noise, we get a spread of data that looks much more reasonable in terms of matching what we see in the true data. So, right, let me actually motivate this next bit. So, you might think that, I mentioned that when you look at this data initially, the data points at week 2 look a little surprising, right? You'd almost want to, if you had more data, you might want to almost say they're outliers. Like these 2 points are pretty close together, these 2 points are pretty close together, and these two seem kind of far apart. So maybe something funky happened on week 2. So in general, we often worry about outliers in data skewing our results. And one way to handle outliers in a somewhat smooth way is to use robust likelihood functions. So rather than using a Gaussian noise model, we can use some distribution that puts more support on the tails. One way of thinking about this is the model is like less surprised to see those outlying values, and so they have less effect on the model fit. One class of nice distributions that we can use are student t distributions. So these have some parameter called the degrees of freedom. If this degrees of freedom goes to infinity, you actually get a Gaussian again. So if this degrees of freedom is small, that means there's more sort of outlying points. If it's large, your data looks more Gaussian. So one of the extensions I have that you could do is to actually try and fit this parameter. But for now, we'll just set it to some sort of intermediate value, 3. It usually seems to be pretty reasonable. And nothing else had to change, right? I didn't have to change anything else about the model. I just switched in the student t instead of a Gaussian instead of a normal. We can fit this model again. And again, the inference code wouldn't have to change at all. And one of the interesting things that happens when you do this is I'm showing here what the posterior mean and standard deviation is on the guide score. And the posterior uncertainty actually goes down when you use the robust noise model. And that's because the model, those somewhat outlying points are less surprising under the student t. So I showed you fitting this model on just the data for one guide RNA, and I said we have like 50,000 guide RNAs. So maybe we could use the data from all of the guide RNAs together to do a better job at fitting the model for each individual guide RNA. And this concept gets called various things, but sharing statistical strength is one of them. And this makes a lot of sense to do here, especially since we're asking a lot of this model right now, right? Like we have 6 data points, and we're trying to estimate a slope, and we're trying to estimate a noise variance, and maybe we'd also want to estimate that degrees of freedom on the student t. So it's just a lot to do off 6 data points. And a nice way to do this, achieve this goal of sharing information between different somewhat related observations, in this case guide RNAs, is to, well, let's first think about these 2 extremes. So one extreme is we could just fit some independent model for every single of the 50,000 guide RNAs. And that's kind of effectively what I've shown you so far. So no sharing of information, but we have very little data to go on for each individual guide RNA. Another extreme is I could say I actually just fix some parameters to be shared, to be the same across all the guide RNAs. So for example, that noise variance, I could say that's the same across every single guide RNA. But a nice intermediate is to learn a prior distribution on these parameters like the noise scale that are present in each of the guide RNA sort of sub-data sets. And the advantage of this is the model can choose where on the spectrum we should be. So if it seems like the amount of noise for each different guide RNA is very different, then the model can learn that this prior should have a very large variance. But if it seems based on the data that the noise variance is very similar across all the guide RNAs, then the model can learn that this prior has a very small variance. And just to point out, this is a very similar concept to having random effects in meta-analysis. So we're effectively meta-analyzing across different guide RNAs. I'll skip setting up the data because that's not very interesting. So we're going to have a prior on the noise scale. And the most convenient way to do this is to model the log of the noise scale. So this is going to be a mean and a variance on that log scale. And then for each of the guide RNAs, so now we have another for loop. So this is saying for each guide RNA in our data set, we sample a noise scale. And that comes from a log normal with the mean and the standard deviation that are additional parameters in the model. So if this is, once we learn this parameter, that's going to tell us how similar should the noise variances be across the different guide RNAs. And then this bit's not so important, so I think I will skip this. And the main, in the actual likelihood, the main thing we need to change is just that the scale is now going to be the noise scale for the specific. This is just indexing which specific guide that data point is from. So this runs across the full data set in about 40 seconds on a CPU. You can verify that on the on the Colab notebook if you want. It would be faster on GPU. There's a little bit of work to get everything to work on you need to transfer all the tensors to the GPU. So I didn't want to convolute the notebook by doing that. So you can see across the 50,000 guide RNAs, these different guide scores. So these ones that are like large negative values, these are the ones that are really working well. These are all ones that are not working, if at all. And then this red line is the learnt prior distribution on the noise scales across all of the guide RNAs, and the actual distribution of the estimates of the noise scales for each guide RNA. You can see this is a bit narrower than this. That's because the histogram is just showing the mean of the posterior distribution for each noise scale. So there would be a bit more uncertainty that's not represented there in the plot. We have a bunch of extensions that I think for time I will not talk about. We have a neural network that predicts these guided efficacies from sequence. I wanted to mention a few Pyro pros and cons if I have time. Yeah. So you can very flexibly build your models. So I showed you, you can use a normal versus a student t distribution, but there's lots more distributions that you can also use, and they're very well, it's very comprehensive, I would say. You don't need conjugate models, which a lot of previous probabilistic programming languages were limited to. And you can throw in kind of all the deep learning magic if you want to. So anything you can represent in PyTorch, you can include in your model as like a deterministic component. Computational efficiency is really good. Even on CPU, it's pretty good. And obviously, if you're doing bigger data sets, you can move things to the GPU. One thing I really like, and I was talking to Shirem about this earlier, is the models are just Python code. So you can, I didn't show this, but you can just have like if statements in there. So you can have a flag which says if student t flag. And then if that's true, then I'll use a student t. And if not, I'll use a normal distribution. And so you can have a lot of different modeling choices or options without having lots of copies of your model code. Their support is pretty responsive. If you are trying to develop a model and it's not working, then they're pretty good about responding and trying to help, which is really important. I don't know if people have tried to use Stan, but it can be a real pain to install. In terms of cons, handling discrete variables is possible, but more difficult than continuous variables, and it sort of inherits that from the deep learning approaches. It's based around stochastic gradient descent. I'd say the documentation is not beginner friendly. The Stan documentation is actually much better from that point of view. The documentation feels like it's targeted towards machine learning researchers who want to come up with the next latest and greatest thing in stochastic variational inference, rather than practitioners who have an actual data analysis problem they want to solve. But I think it is actually really good for the latter set of people, so I would encourage you to give it a go anyway. I tried to sort of hide this, but dimensions can get a little confusing with complex models. So what dimensions of tensors are corresponding to. One unfortunate thing now in a way is there's now Pyro and NumPyro, which is based on JAX, which is another deep learning library. So that can be a bit confusing. You'll think you've found some function you need, and actually it was for NumPyro instead of Pyro. And then sometimes to get models to perform well, you need to do a bit more work. But honestly, if that was the case, I think it was just always going to be a hard modeling problem. So you can end up, you can make custom variational posteriors that represent some correlation in the posterior. And I mentioned good initialization is often really important. This is honestly not a Pyro thing. This is just a Bayesian hierarchical probabilistic model thing. It's often really useful to give initialization that gets the model in roughly the right place. And yeah, some parts of the API are a bit clunky. I don't know if, where I showed the initializing one of the parameters, it feels like there's a lot of boilerplate you have to put to do that. Further reading, we have a preprint up on both this CRISPR screen that we did and the computational analysis, including this model. The CBAS bit is screen efficiency analysis with Bayesian statistics or something like that. It's not too bad as backronyms go. The official Pyro docs are at pyro.ai. There's a couple of intro to Pyro videos that I think could be fun for this audience, partly because they are both targeted at comp bio. So Justin Hong is actually a PhD student with my collaborator Elham Azizi, but he did this when he was in the Yosef's group. And then this is from some of the actual people who worked on Pyro, and it's a re-implementation of ScanV, which is one of the extensions to SCVI, which probably many of you know. It's one of the, it's the deep learning single cell analysis pipeline. And then, yeah, the Stan documentation is amazing in terms of model building choices. Okay, that's it. And sorry, I'm over time, I think.